to Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom. We'd like to thank our episode sponsor, Spark Cures, for their support of this Myeloma Crowd Radio program. Spark Cures is a myeloma clinical trial finder that you can find on the myelomacrowd.org website, and is a wonderful way to easily find clinical trials. We have with us today Dr. Martin Kaiser of the United Kingdom, and we are here at ASH together. So Dr. Kaiser, welcome, and thank you for joining us. Before we get going, let me introduce you. Dr. Kaiser is a clinician scientist at the Institute of Cancer Research and honorary consultant hematologist at the Royal Marsden Hospital in London. Dr. Kaiser attended medical school and obtained his MD degree in myeloma research at the University of Aachen, Germany, and completed his specialist training at Charité University Hospital in Berlin, Germany. In 2011, he joined the Institute of Cancer Research, or ICR, and since 2014, Dr. Kaiser has headed the myeloma group at the ICR. His research focuses on integrated molecular profiling for biomarker and target identification for stratified medicine in multiple myeloma. So he works on specific areas of activity like genomic and epigenomic molecular profiling of high-risk myeloma and epigenetically driven disease. Through collaborations with the ICR's Division of Cancer Therapeutics and external partners, the group also explores novel and innovative tool compounds and candidate drugs for preclinical and clinical drug development in myeloma. Dr. Kaiser heads trial-specific myeloma biobanking for multicentric UK Myeloma Research Alliance trials and for studies in the Myeloma UK Clinical Trials Network. He's chief investigator of academically led trials with a particular focus on genetic stratification and biomarker discovery. In collaboration with Dr. Messiot of the Royal Marsden Hospital in Radiology, Dr. Kaiser is investigating innovative imaging methods for improved patient care, in particular diffusion-weighted whole-body MRIs. Dr. Kaiser is a member of the UK NCRI CSG myeloma subgroup, is an elected member of the executive board of the UK Myeloma Forum, and lead for the Genomics England GECIP hematology subgroup for myeloma. So Dr. Kaiser, we are happy to have you and look forward to our talk today. A lot of excellent research has come out of the UK, and there's a strong patient advocacy group as well in the UK. Could you give us perspective of treatment of myeloma in the UK?
Yeah, I think it is, of course, I see it as a big advantage of having seen how different systems work and how they might impact on patient care. I think in some sense, the way how I trained and how I'm working in the UK, of course, there are a lot of similarities because both are healthcare systems do define the drugs that are available for a broad population effectively. I think what makes it a bit more special in the UK is that there is a attempt to have a patient pathway in terms of what the different lines of therapy are that can be given, and that has both up and down sides. There's differences in treatment options, and I think they're along the patient pathway. I think there is a clear availability of very effective treatments at each stage of the disease, but it is not necessarily a free pick that you can choose whatever you want, but there has been, of course, data produced as part of clinical trials, and that leads to a approval that is really for a certain indication and a certain point in the disease where this type of regimen can be given. That still means that, of course, we are doing personalized medicine, we are adjusting the doses, we are looking at what the patient wants, we're taking all aspects into account as the personalized medicine that doctors have done, ever since, effectively, which is a different type of personalized medicine than the one that is now we're entering into with molecular stratified treatment. All of these things are still going on, but it nevertheless gives more of a clear sequence of what can be given at which time point. And I think there are certain elements of that sometimes make you want to be probably a bit freer in your availability of what you can pick. But on the other hand, once a drug is approved as well, a patient can really access it without any barriers. And I think that is a big aspect. It's just something that's so deeply built into the UK's healthcare system that once a drug has been through the approval process, it is truly available. There are no ifs and buts. There are no, other levels anymore of intricacy that sometimes can mean that a patient can have a drug, for example, as an inpatient, but not so well as an outpatient or the other way around. And I think that's what we're often facing in other healthcare systems, that there are a slight difference. I think what is good in the UK as well is that it is really considered a joint resource, and there is a constant process going on to look at how to best use the resource. So oral agents are always favored over very difficult to administer drugs, for example. It's not saying that these other drugs, if they're very efficacious, they're still given, but I think it is very much in the patient favor as well that, it's looked at things that are easy to administer, easy to take, easy to give the patient effectively a lifestyle as well that is as compatible with all the challenges that we're all facing in our lives all the time as possible, whereas sometimes in other systems there might be an incentive to actually rather favor intravenously given therapies, despite them probably being equally as efficient as an oral drug, for example. So these can be advantages as well. I think I'm very lucky to have experienced both types of systems with their slight differences, but I think I'm very glad that we can give patients the care and the access to drugs most of the time, although we always want to have more available and follow on with the new developments, of course.
Now I have a question about clinical trials. I know European clinical trials are different from US clinical trials. In France, for example, or other countries, patients might go on to trials that the experts have constructed that are really large scale. Is that the case in the UK? Now they have very high participation rates in their trials, but it seems there's less variety in the trials. And in the US, there's more variety but more difficulty accruing. So there are sort of positive and negatives on both sides.
There are different aspects. I think some of it has to do with that there is a public healthcare system that has been designed from the beginning as having an inherent research aspect embedded in it. So this is probably a right assumption by many patients that they do understand that public healthcare system can only work and evolve and actually produce novelty if they participate in trials as well. And in some circumstances it might give them as well an advantage in having access to a drug that's not available on the public healthcare system, but most of these trials are randomized. So in the end of the day, we can never guarantee that the patient will get access to the new drug. So in some sense, I think it is deeply embedded as well that there is a clear desire or a clear willingness to really contribute to the progress. Nevertheless, I think there is an aspect, of course, that if you have a healthcare system like in the US where drugs are probably approved faster, or the approval often comes first through the FDA and is then free to prescribe by any physician, there is of course more of a choice, and that can just mean that people might not want to consider going into a trial where they actually, but it will be a random assignment to the one or the other drug.
Let's go back, because you mentioned the oral therapies, and those options seem to be growing in myeloma. What's your opinion about the move from doublets to triplets, and also new options patients may want to consider?
I think it is great that increasingly more oral agents are available, and luckily really most of the oral agents that are around are really well tolerated, which is, one has to say, in comparison to other tumor types, luckily for many of the drugs that we use, true, but I think it is particularly still a really good feature of that many of the drugs. There is so much experience now with them, or they actually have such a biological background that they can be taken and tolerated for the intensive periods of treatment, but also for maintenance type of therapies, and that's very important in myeloma. The most important use, I think, of oral agents is really in the maintenance setting. That doesn't mean that I don't think that they should be taken in the upfront or induction treatment settings as well, but I think that is really where they make such a huge difference, because it can be many years now, luckily, that patients stay in a remission on a maintenance therapy, and not having to come to hospital that often is making a huge difference. So we have, at the moment, Revlimid, for example, approved as a third-line treatment drug in the UK, which is as such great in that we can use it for as long as we want in that setting. And we do have a lot of patients that are having ongoing benefit and continuous benefit from taking this maintenance drug. We're hoping that this will probably move into earlier lines of treatment. And here at this meeting, Graham Jackson has just shown the results from the large Myeloma 11 trial that was run in the UK, and that provided really elemental evidence for Revlimid maintenance therapy, particularly after transplant. It was a large trial, and as we spoke about earlier, many patients in the UK were really very happy and willing to go into this trial, and I have treated a lot of them myself as well and have seen them benefit from it. So I'm not surprised that, especially in the transplant setting, there is now a demonstrated overall survival benefit as well. So really a long-term effect of patients going onto this maintenance treatment. The first trial that on its own, as one single trial, in a predefined setting really found this overall survival advantage. There has been a recent meta-analysis that is completely in harmony with this finding now. So I think that is a very important finding, a very important case for this type of treatment after autologous transplant. We do see that other agents are being tested in that space at the moment as well. There are the oral proteasome inhibitors, for example, that are particularly ixazomib, which has been tested, and I'm very much looking forward to the outcomes of the phase 3 trial. Again, another trial where a lot of patients from the UK participated as well. I think that is really a very important space for the oral agents. In the different phases of induction treatment, we are trying to use triplets as well. This is where, particularly in the healthcare system, it is not globally possible to use two novel agents together. We do have an induction therapy with Velcade, thalidomide, and dexamethasone available, and that's very effective actually. But when it comes to combinations, for example, like Revlimid and ixazomib, at the moment at least, that is not possible. This might become available on the healthcare system, which would be great as well. I think is another very effective regimen. But I think really a very big impact is the maintenance setting.
I agree, and I'm hearing that everywhere. Let's talk about clinical trials you have open and how patients can find UK clinical trials.
Clinical trials in the UK are often registered on official portals, if you just mentioned them. There is also a very active patient information organization, Myeloma UK, who does keep a register of sites that are participating in clinical trials, and that's another point of contact. We do have a very closely knit community in hematology specialists in myeloma as well, so clearly their treating physician will be very happy to keep out an open eye for whether any trials that are not available at the local center are actually available relatively close to where the patient lives. The treating physician will be very happy to advise the patient on where a trial is potentially open. So distances are not so long normally and not so far in the UK. In terms of trials, it is really a unique setting that the healthcare system does have an inherent research aspect. So there are a lot of academic trials going on, and that is brilliant. So there is a long tradition of having trials for the newly diagnosed patients. We are now at the moment in a phase where the large myeloma 11 trial has finished recruitment a certain while ago. We're now seeing really the amazing outputs of that trial and the amazing results of that trial, which will hopefully lead to really knock-on effects in terms of what is available, what's possible in terms of treatment. We have then a number of trials that really tests concepts and that are trying to push the boundaries. These are run sometimes in the newly diagnosed population, but often as well in the relapse refractory population, and many of them are run again with Myeloma UK, which has been a very early on understanding organization to actually contribute to set up clinical trials. So it interacted very early on with the NHS embedded research infrastructure and kind of made a case for myeloma patients in particular to actually access certain aspects of that infrastructure so that it is easier to set up a trial and to find the collaborators that are always necessary, for example, in pharma, to get really novel drugs, novel combinations to patients in the UK. Of the trials that are currently running, we have just started a trial for high-risk patients. This is really a first, and I think it's in its scale, not only a first in the UK, I think it is internationally quite a unique design and quite a novel setup. So we have designed a fairly big study because we wanted to really make this impactful and have as many patients benefit from it as possible. So we're going to screen 600 newly diagnosed patients. That is big, and we're really lucky that we could draw so much experience from our genetics work that we did before on the large myeloma 11 trial, which in total included 4,000 patients, more than 4,000 patients. So that we had really solid data on how really to best identify patients that with current treatments unfortunately are not doing so well and that could benefit hugely from having more innovative approaches and really getting access earlier to the novel drugs that are clearly showing very much benefit in a relapse refractory setting. So there is little doubt that they will have a big impact in the newly diagnosed setting as well. So the design is that these patients do give their bone marrow, which is sent to our central laboratory at the Institute of Cancer Research, to be then immediately worked on molecularly profiles. We do tests for genetic aberrations. We're using a molecular test that is consisting of a method that's called MLPA. It's a difficult abbreviation actually, but the concept is that we can test the copy number status of different regions. So whether there is actually more genetic material or less than there should be in the cancer cells very rapidly for many samples in parallel. In the old days, or until recently, or in many centers still, the standard method is FISH, which is a very good method, and I think there is no reason to be critical of the result as such, but it is just a method that has a certain maximum number of samples that a human can just process. There is no straightforward automatized process of analyzing the data. That's why we have actually analyzed pretty early on this MLPA method, because we had a set of patients from earlier clinical trials, again, this is really at the core of everything that we do, that had FISH data, so we could easily compare whether this new method is actually exactly giving the same results. And it did, so published that as well. This has fed back as our experience now into running this trial. Also, we had another method which is effectively a PCR, which looks at whether genes that are involved in translocations are highly expressed. So whether these genes are having multiple copies of the genomic region read out. And if that is the case, we know that from earlier work, for example, from Leif Bergsegel's group, that shows that there is a genomic change has happened in that region effectively. So we can call translocations with that. That's the one side. So we actually test for the T414 translocation, the T1416 translocation, deletion of 17p, and gain of 1q. And we have found before that these lesions, unfortunately all of them do confer a risk of that the disease comes back earlier. But in particular, if two of them happen at the same time or are found at the same time, the risk is even higher. And if you have a treatment that could potentially mean more effort for the patient to come more often, you want to make sure that you're treating the right patient. You're not treating someone who might have had effectively a very similar disease history from there onwards, but you want to make sure that you really serve the patients most that have the biggest need. Another method that we're using is gene expression profiling, and I'm sure many of the listeners will be very familiar with that. This is something that has been probably more broadly applied in the US for some while. There is now standardized tests in Europe as well that have been developed by the Dutch group originally. That is the Skyline assay, and that is a standardized assay, so we can implement it, or we have decided to implement that into this trial as well. And that was again coming from data that we analyzed in the large myeloma 11 trial, where we actually looked at genetic risk and gene expression risk. And we were curious, are these actually the same patients if you apply the one method or the other? Because there were very few, or actually virtually no data around of people that had really looked for both of them. So we looked for patients that had these two aberrations genetically, which we also internally at least term double hit, and we tested the same patients whether they had a genetic high-risk marker, gene expression high-risk marker. And we found that some of them were identified by both methods, but there was a subset of patients that was only identified by the gene expression high-risk marker, and another set of patients that was only identified by the genetic double hit high-risk feature. So we decided actually that it would be absolutely adequate to include all of them, because we do see that they unfortunately all have a shorter duration of remission with current treatments. And if one applied only one or the other method, one would probably miss patients that are really in need. So we were, on the one hand side, of course, puzzled by this finding, and we still are working very hard on understanding why two different methods identify different patients, but the pure fact that they all are in need of better treatments made us, of course, very motivated as well that we can use both methods together to confer the biggest hopeful benefit for the patients by enrolling them into this trial. So we're testing this, we're feeding the result, whether this was found, back to the patient, and if it has been found, we are offering them participation in the trial where we use not only a triplet but actually a combination, in the beginning at least, of 5 drugs.
Oh, wow.
So we use a combination of cyclophosphamide, Revlimid, Velcade, daratumumab, and dexamethasone.
Okay.
But we do acknowledge that that is, of course, a relatively intensive regimen. What we want to make sure is that we really offer the patient the best treatment in the very early days of the disease. What we want to avoid is that any resistance mechanisms are happening early on. We want to give the patient the chance that the disease is responding as quickly and as best as possible. We do, however, very clearly think that the personalized medicine that doctors have always done is a very important element. So the trial is not strict in the sense of that the patients have to carry on at the same dose or at the same schedule, but we have built in that the physicians watch very closely how their patients are tolerating this treatment and are reducing drugs early on, because what we want to avoid is that patients have to stop treatment. Effectively, we offer everything, but it's more like an offer, and the physician then has to, in the dialogue with the patient and observing the patient, decide how to adjust treatment so that it's best tolerated and at the same time offers the most benefit, actually. So these patients then undergo an autologous transplant as well, and after that the drug doses and certain drugs are just tapered off, because we do want to keep some or a lot of pressure on this high-risk disease clone in the bone marrow and really push it down to lowest levels. In this trial we are measuring central MRD, which is another brilliant feature of the UK research group, is that we have a very collaborative structure. So we have the genetics laboratory where I'm doing the work and my team is doing the work on the genetic profiling, but we're working very closely with a lab in Leeds, and that is led by Roger Owen, and Ruth Latoot, who is the scientist in his lab, is going to present data at this year's ASH as well, again from the Myeloma 11 trial. But these guys can measure MRD really perfectly. They have the experience of thousands of samples having measured as parts of trials. So measuring this in the high-risk population as well will allow us in this trial, for example, to have earlier readouts. We can take that measurement at certain key time points to see whether patients respond better than patients that we have profiled before from the trials that we have worked on in the past. So we have an immediate comparison, actually, to patients that have received also very effective treatments, but down to their disease biology, probably were not benefiting as much as other groups, and we can immediately read out whether this other treatment is actually benefiting them more, and that will feed into further developments of future trials, for example.
We're hearing more and more about MRD testing as a potential new endpoint for clinical trials. The UK sounds like you have the advantages of genetic testing, MRD testing, research, patient advocacy, and that's just fantastic.
I think we're very glad. I'm very happy, each day virtually to be able to work together with so many great people in this collaborative, and of course for the patients that they want to take part in this research as well. And I think with this trial we hopefully have designed something that really brings back a lot of the innovation and the insights that we have won through, of course, previous patients' commitment to giving, for example, bone marrow material, but actually really benefiting the next generation, because we're applying a lot of the methods that we have developed back onto the patients or for the patients that are enrolled and taking part into this in this trial now.
Mm-hmm. That's great. What a wonderful collaboration. How many hospitals are in the network?
It's been a lot of work. It's always difficult to set up trials like this, and one goes to endless meetings and sometimes comes out and thinks, well, maybe this is stuck now, and maybe we're not getting anywhere, but it just pays out at the end. It really pays out the moment that you get something off the ground. So for this trial, there will be at least around 20 hospitals, but we're looking to extend this trial because, of course, it's understandable. Patients are very interested in this. We want to really give everyone a chance to be considered for this trial. It is a lot of hospitals. We have started with the biggest hospitals, and that is really a very large catchment area if you count them together already. For a large trial that ran over many years, like Myeloma 11, the number was even higher. So in total, 100 hospitals took part in this trial, which meant that really virtually every patient had a chance to take part in a trial like Myeloma 11, and that's the only way how, in the end of the day, you can really get such a huge trial together that enrolls over 4,000 patients. I think another aspect that was very important about Myeloma 11 is that it is a trial that is really for all age groups, and there is particularly a group of patients that are close to 80 or over 80 that in reality are a large group of patients with myeloma, but in the normal trials they're not that represented, and I think it has to do with lots of factors as to how easy the treatment is, whether it's oral, whether it's not oral. Myeloma 11 had the clear advantage. It was a full oral regimen, and it was rolled out in really every hospital around the country. So it has a very large proportion of these patients as well, and I think this is something where a lot of insights will be won. There are already data shown here at ASH that help us understand better the needs and the specific challenges that these patients are facing, whether we can identify markers in the blood, relatively simple markers, for example, that could tell us, look, this patient might be running into trouble just because generally they're not as fit as someone else, for example. And I think we will hopefully be able to derive certain models of care for this type of high-risk patient as well, because that's another type of high risk.
Right. If people have a pre-existing condition, that prevents them from getting on a drug. That's a high-risk feature in itself. Being as fit as possible to be able to receive some of these treatments is really key.
Oh yeah. No, that's very important. And I have to say, it's a conversation where I really constantly try to motivate patients as well. And I think it's a very important aspect. It's very important to make clear that it doesn't take much. It is often about just being out for, or some way active, mild activity. It's doing it regularly, even if it's a long walk, regularly is fine. Keeping, some eye on your diet, not to just have one type of food, but actually have fruit, vegetables in there. I think it's a question that naturally comes up all the time. It is often a question that comes up in the first conversation that I have with a patient about when the disease is diagnosed. What should I do food-wise? And I think it's very important to understand that it's not a disease that is caused by any wrong behavior. It's not anything where an environmental factor plays a big role. We know that from large studies. But it is, of course, something where, as you rightly say, keeping yourself fit and healthy just generally has so many good features in terms of tolerating drugs better, tolerating your treatment better, knowing yourself better as well, understanding when are you really unwell, when should you go to a hospital to actually be looked after, and when is it maybe just your bad day, exactly.
Yeah, not myeloma related.
Yeah.
Or could you go to a transplant even if you're older? Or
Exactly. I mean, I think we have learned a lot of lessons about that absolute age is not really the barrier for a transplant. We do transplant actually quite a number of patients that are above the age of 70. If they are fit, if they want that, we have a very open and frank discussions with patients. But absolutely, there are more and more people around that are very active and fit. Being old is not a feature of having a certain age. It's actually probably more a feature of whether you depend a lot on someone else's help.
I agree. Those frailty scores really segment people into fit, unfit, and frail categories. So if you're high risk, you need to be as fit as you possibly can be.
Absolutely, that's absolutely true. And one has to say, of course, the background of the disease, or how it's actually working, which we call the molecular feature of the disease, but that in some sense always shines through. It is a feature still that mandates us to be more on the case and to really give the right treatments for the patient, so that we can control the disease when there is a risk of that it could come back earlier.
It's an exciting clinical trial. Are there others you'd like to talk about? I heard of a CAR T trial in the UK, but I don't know where.
There is academic centers that have developed CAR T cell trials. For example, at University College, there is a CAR T cell trial of a construct that was developed there. But of course, we are getting as well now, after the initial development was done in the US, we are getting now trial programs as well from the CAR T cells, that some of them have already received breakthrough approval by the FDA, for example. And I think that's going to be a big benefit for our patients, of course, as well. An important option. I think it's an option that we can offer because transplant and transplant centers have so much experience in the UK and doing quite complicated procedures with a lot of patients already. So I think it's a great country as well from a development standpoint and from clinical trials, that we can hopefully offer CAR T cells very soon if they mature further in their development pathway to a lot of patients.
How do you see immunotherapies in general progressing, like the BiTE antibodies, the antibody conjugates, the monoclonal antibodies and vaccines? How do you see that rolling out in the United Kingdom?
We're starting already. I think we have also been huge contributors as a whole country to the daratumumab, for example, development programs, as well as the elotuzumab development program before. There are lots of UK patients who are actually treated as part of these trials and have contributed a lot to the outcomes of these trials, all of which are very exciting. I have to say, this is a real amazing development. It's absolutely great that the immunotherapies are coming along. I think the important aspect that they carry with them, at least some drugs that we know now already after phase 3 trials, their feature is that they have a very good safety profile and a very good side effect profile that makes them pretty tolerable. And I think that opens just the spectrum as how they can be used, both in patients that may not tolerate that well because of frailty, for example, certain other drugs, but also in terms of the possibilities of combining them with lots of other drugs without adding yet another level of significant toxicity, for example. I think this just gives so many more options. In terms of the new developments that are coming on of the drugs that don't have approval yet, that are going through the early stages of development, I'm very sure that the UK, again, will be a very keen participant as a whole, as a community in these trials. And I think there is an understanding as well in terms of the regulatory authorities in the UK that these drugs are fundamentally different. They're undergoing review at the moment, some of them at least, and we hope that we see them really being available in the near future. It's always impossible to predict. There are many factors that are playing into that, but I think the basic understanding is there, so we're pretty hopeful.
Well, I'm very grateful you're participating in all these trials and making the data come out so decisions can be made. My last question is, what are the most exciting things you're seeing here at ASH?
I think some of the immunotherapy approaches are really very exciting. The ones like BiTE antibodies, like CAR T cells. And what I'm looking for now here particularly is the really impressive efficacy, and particularly if it's combined with a good tolerability profile. I think that will just make it so much more easy to find combinations that we can use together in the future, that we're not hitting a ceiling of adding yet another drug on another that all cause the same side effects and make patients' experience effectively not as good as it could be effectively. So because it is a disease that I think does need these strategies of some form of continuous treatment. With that, we have managed to really transform outcomes for patients from, unfortunately, really short times of remission to patients staying in remission sometimes for 10 years longer or longer. So that's a real goal. We do want to transform more the outcomes for our patients that have diseases that tend to be coming back earlier. We're working very hard on that. I think we have made good progress on that side as well. And the better understanding of the molecular makeup of myeloma and how it really works is very clearly very deep, close to my heart. So there were a lot of exciting talks about that here at ASH as well, which would take yet another session to really go into depth about that. But I think we have seen a lot of technological breakthrough in how we can analyze the genomes, the epigenomes of the myeloma cell. We're working very hard on that. It's very exciting to see other groups are making progress on that front as well, and I think that will be a big area of collaboration in the near future, that we can put all the pieces of the puzzle together to really understand how this disease works, to get yet the next level of therapeutics that are hopefully as effective and more as the ones that we have already.
I've seen a lot about driver or resistance mutations. Maybe these are all pieces to the puzzle. If you overcame resistance, you could give someone 10 or more years of life.
Absolutely, absolutely. I think there is a lot of more effort going on now to really understand how the disease evolves, for example, if it relapses. And really, again, through the clinical trials that we have run, we have a lot of patients who gave bone marrow at different stages of their disease. So this is an analysis where we are really focusing on at the moment to effectively look at how the tumor changed over time, not only in a handful of patients, as it has been done very impressively in the past, for example, by Jonathan Keith's work, but we want to look at it at a scale of really several hundred patients now, and we are in that position to understand more networks. In the end of the day, we have, I think, more insight into that the myeloma cell is not simply built. There isn't just one engine in the whole process, but it has a whole network of different drivers, and understanding how they work together, or actually whether there are subgroups in which one whole network is much more active than another one, and then actually going with the right drug for that subgroup is going to be really the ongoing challenge for the coming years. I think it's not as simple as that you will have one golden bullet for something, but we do see now these networks emerging. We do see more that the pieces of the puzzle are fitting together into certain categories, and that's really what we're working very hard on.
Well, we appreciate your work, because once you start defining those categories, then you can start treating those categories differently, which brings us back full circle to personalized medicine. Well, thank you for all you do for multiple myeloma patients around the world, and for joining us on Myeloma Crowd Radio.
Thank you very much for having me. Thank you.
Thanks for listening to Myeloma Crowd Radio, and we hope you join us next time to learn more about myeloma research and what it means for you.
It is Ryan here, and I have a question for you. What do you do when you win? Like, are you a fist pumper, a woo-hooer, a hand clapper, a high-fiver? I kind of like the high-five, but if you want to hone in on those winning moves, check out Chumba Casino. At ChumbaCasino.com, choose from hundreds of social casino-style games for your chance to redeem serious cash prizes. There are new game releases weekly, plus free daily bonuses, so don't wait. Start having the most fun ever at ChumbaCasino.com.
No purchase necessary. VGW Group. Void where prohibited by law. See terms and conditions. 18+.